Hello lovelies, in this video we're going to be looking at the control of gene expression for your A-level biology. Now we need to be really specific on the details of this, so take it slowly, write down things carefully. Once you finish this video you can pop over to the website and look at the question the doctor wrote, which is written you specifically about this video to make sure you thoroughly understand everything.
Transcription factors. So a gene is expressed or switch on, which is what we mean by expressed, when it's transcribed into mRNA and then translated into a protein. So you have to go through that whole process for a gene to be expressed, i.e. to turn a gene into a protein. Only a few genes in a cell will be expressed at any one time, and as we've looked at, depending on the cell, all of the genes could be expressed or only some of them can be expressed. So let's have a quick recap and reminder of what transcription and translation looks like. So transcription of the gene in the nucleus uses RNA polymerase and it converts DNA into mRNA. That mRNA is technically pre-mRNA because then it needs to be spliced in order to remove the introns, leaving the exons behind that then get stitched back together to produce a piece of actual mRNA. The mRNA then leaves the nucleus via the nuclear pores, and remember it's obviously single-stranded. And then that mRNA binds to the ribosome, could be on the rough endoplasmic reticulum, or it could be in the cytoplasm. And that ribosome and the tRNA molecules used to then translate that mRNA into a polypeptide chain. So a chain of amino acids forming a protein. That protein will then move to the Golgi and it'll either be modified or packaged and sent somewhere in the cell or secreted or sent to the cell membrane, whatever type of protein it needs to be. So that is transcription and translation. So if we are going to stop or edit or control transcription or translation or control the expression of genes, we're going to need to edit or have a step that we're adding to these stages in order to either stop it or promote it from happening. And that's what we're going to look at today. So we're going to look at transcription factors and how they affect transcription. And we're going to look at interfering RNAs because they can actually affect translation. And so both of these can affect whether a gene is expressed or not by affecting the different stages, either transcription or translation. Okay, so before transcription can begin, a gene needs to be stimulated or activated by a regulatory protein, which is called a transcription factor. So our definition for transcription factors are that they are proteins which travel into the nucleus and control the rate of transcription. So either by activating or repressing RNA polymerase or the action of RNA polymerase. So each transcriptional factor will bind to a promoter region on the DNA. So it's a section of the DNA at the start of the gene that's going to be transcribed, and it's called the promoter region. And in here you can see it's got this kind of green box around it. It'll be a specific sequence at the start of pretty much every gene that is where RNA polymerase knows to bind, and also the transcription factors know where to bind in order to start the process of transcribing this DNA. Transcription factors then bind to this section of the DNA in the promoter region, and then that binding is either going to block or promote the action of RNA polymerase. So you'll see here we've then made this complex where we've got our transcription factor, we've got our RNA polymerase, and we'll see the RNA polymerase is doing its work and we're producing that mRNA. Transcription factors can either activate or block the action of RNA polymerase. So we call transcription factors that act as repressors. They are repressors, they prevent the RNA polymerase from binding and therefore they stop transcription of the gene. Or we have activators or promoters which bind and then allow the binding of RNA polymerase and therefore trigger the transcription of the gene. We need to learn a specific example of how transcription factors work in prokaryotes. This is called the lac operon. In prokaryotes, transcription factors bind to operons. An operon is a cluster of structural genes that are all transcribed together. It often includes control elements and regulatory genes. Structural genes code for useful proteins, such as enzymes. Control elements include the promoter, where RNA polymerase binds, and an operator, which is the binding site for transcription factors. Regulatory genes code for an activator or a repressor. An example of an operon we need to know is the lac operon. E. coli bacteria can respire using lactose if glucose isn't available. So they have genes that code for the enzymes needed to break down lactose on an operon. The operon includes 3 structural genes, LAC-Z, LAC-Y, and LAC-A. The transcription of these genes is affected by the presence or absence of lactose. When lactose is not present, the regulator gene produces the transcription factor R, which acts as a repressor. This binds to the operator site and prevents RNA polymerase from binding, so no transcription occurs. When lactose is present, lactose binds to the repressor R. This causes it to change shape so it can no longer bind to the operator site. This allows RNA polymerase to bind to the promoter region in the operator site, and all 3 genes can be transcribed. This allows the bacteria to now break down lactose and use it as a substrate. So, transcription factors can also be regulated, which is adding another layer of complexity, but basically transcription factors can be stopped by inhibitor molecules. So they can be stopped from binding to the promoter region. Inhibitor molecules can bind to the transcriptional factor and prevent it from attaching. So without the transcriptional factor, the gene may not be transcribed if the transcription factor is an activator, or the gene may then be expressed or switched on if the transcription factor was a repressor. So let's look at an example because that sounds a bit counterintuitive, but if our transcription factor is a repressor transcription factor, normally transcription wouldn't happen, it would be stopped. So no transcription of this gene is happening. So RNA polymerase can't bind, it's blocked by that transcription factor being there, so we're not going to get any expression of this gene, we're not going to make a protein from this gene. However, if a transcription factor inhibitor comes along that inhibits that transcription factor, prevents it from binding to the DNA, then now RNA polymerase isn't blocked, and so the transcription occurs. So a transcription factor inhibitor, in this case, if the transcription factor is a repressor, causes the transcription of the gene, and we will get mRNA, and therefore hopefully we should get a protein as well. The opposite case to this is if your transcription factor is a promoter. So the binding of the transcription factor normally promotes the transcription or activates the transcription of the gene. Then you will get a gene product, you'll get mRNA produced, and we'll have hopefully a protein translated from the mRNA that's been transcribed from this gene. But in this case, if we have a transcription factor inhibitor, then we're inhibiting a promoter transcription factor. So that means that we won't get transcription this time. So without the transcription factor there, RNA polymerase won't know to bind or won't be activated to bind, and so no transcription will happen. So if we inhibit a promoter transcription factor, then we don't get any transcription of the gene, and the gene is turned off or switched off. If we inhibit a repressor transcription factor, then the gene is turned on or expressed. Okay, so you will be given context in an exam. You'll be given either a diagram or an explanation of an action of a transcription factor or its inhibitor. And so you will be just applying this knowledge that you know, and you've heard about transcription factors. You know they can promote or they can repress. And then you'll be kind of given the context around it and sort of asked to figure this out using your knowledge. So far, we've been talking about how we can prevent or control transcription. We also have to think about what happens after transcription and how we can control the product of transcription, so mRNA, and how this can change then how that mRNA is expressed into a protein. So first of all, primary mRNA, that is directly taken from the DNA code, contains both introns, which are sections of DNA that do not code for proteins, and exons. These are sections of DNA that do code for proteins. So these two things are all kind of together in the long stream of mRNA that we have produced. And what we need to do is we need to remove those parts that do not code for proteins. So we need to remove the introns. The introns are removed or spliced out, or this process is known as splicing, to only leave the exons behind. We remember this by saying the introns interrupt the exons. So they're the ones that have to go. So we remove our introns, and then we're going to stitch our exons together. Endonuclease enzymes are involved in this editing and splicing process. This splicing can change, obviously, what then gets translated into a protein. So some genes can be spliced in different ways to give a different sort of sequence of exons, and therefore that gives a different outcome of what the protein will actually be like. The introns, although we said they don't code for proteins, they usually control the regulation of other genes. So they're still important and they still have a role, and it's mostly to do with controlling other gene expression. Another form of post-translational control is through phosphorylation and also the kind of the process that we've looked at with that secondary messenger model. Not all proteins function straight away after being translated. Some of them need to be activated. Activation can occur through phosphorylation. Hormones and sugars can control protein activation by activating the secondary messenger, CAMP. We've seen this before when we looked at the effect of adrenaline. CAMP goes on to activate proteins inside the cell by phosphorylating them. This changes their tertiary structure. For example, it can activate pKa, or protein kinase A. CAMP binds to pKa and it releases active subunits and removes inactive subunits. pKa is now active and it may go on to phosphorylate other proteins. These could be enzymes or they could act as transcription factors and so therefore affect changes inside the cell.
Ouch! This is why in some videos I'll explain scratches.